moved to do graduate work at University of California, Santa Barbara, and then she did a postdoc in the same group where I did my postdoc at University of California, Irvine. So we worked side by side for a while in a very productive collaboration. And then she moved to University of California, Merced, where she's now a professor, an assistant professor at the moment. And so her graduate work was on terahertz absorption spectroscopy and circular dichroism. And that was both on water and salt solutions and on proteins in solution. And then she moved from there to work on molecular motors because they captured her imagination, like they do a lot. And she has stayed with that ever since, and so she's today going to tell us more about multiple motors working together, which is a very interesting topic for biology, but also in bioengineering and related subjects. So without further ado, please welcome Jing.
Thanks for having me here. Can you hear me okay? Sometimes my volume control is not quite there. So just wave if I trail off. So so I'm from University of California, Merced. Merced is in 2005. It showed up as a new campus. Here's the relative location. Let's see. So my talk today is on the ensemble function of molecular motors. Ensemble function as physicists is many-body problem, and molecular motors is really what I feel very privileged to work with. So what is molecular motors? Let's say my brain was washed when I was undergraduate in a hardware instrumentation lab. I consider myself as I identify as a hardware instrumentalist. And here are a list of physical machines. Not quite sure if you can see. Physical machines that have different forms. Some of us interact with them daily. Some of us on different frequencies. And one can argue that despite their relative, their differing complexities, here's a free electron laser at UC Santa Barbara, and here's the iPhone 5, which I think we all own one version or another of it. Here's the optical track, and here's the express machine and car, and so on. All these have components that we recognize. At the heart of it, even this iPhone 5, if you take it apart, it's going to have some resistors, some capacitors, some batteries, and so on. And same thing here, you can see it very clearly. And to a large extent, we understand what these building blocks are, and reasonably well how they function individually and how to put them together for our physical machines. So I'm going to show you a version of a biological machine that, and I will argue is rather beautiful and fairly robust. And I'm going to ask you the question, what are the building blocks of these biological machines?
Nowadays it should be an ice.
All of you have answers to that, and I had no answers until I graduated from graduate school. That is, I now understand that there is one type of building block for this biological machine that is called molecular motors. And here is a simplified cartoon animation of a molecular motor, this floppy thing carrying a cellular material, this blue thing walking on a track. It's kind of a cellular scaffold. And just does this, really just two conformation change, A, B, and keeps on going.
What's the driving force?
The driving force, so excellent question. It's stimulated by chemicals.
Is it a concentration?
So it's you can tune the concentrations of the chemicals. It's called ATP. It's a triphosphate. As we all know, we break apart chemical bond, it releases energy. Triphosphate goes to diphosphate, and it releases about 20 kT. And this guy, each step takes about 5 to 10 kT.
Is there any directionality to this, or is it just random?
There is.
Okay,
What drives the directionality? That's a huge question. We don't actually know. So biochemists are actually chopping pieces apart and say maybe this section of the motors, we call it molecular motors, maybe this section is responsible for the directionality, and then glue together these Frankenstein version of molecular motors and see whether or not those versions have directionality. To a large extent, we don't know the precise mechanism yet, which really makes it very interesting. Not only that it has directionality, this is one type of motors. There's another type of motors that is sort of canonical inside of our cells. This is scaffold has structural polarity. It's not electrostatic. It's not polar polarity, but structural polarity. This motor walks one direction, and another type of canonical motor walks predominantly in the other direction, although it backsteps as well. So a very rich set of questions that hasn't been fully understood. Then you ask what happens when you put them together. Fine. So the point I want to make in this slide is that it's a key building block of biological machines. It's universal to all humans. It runs in every single cell of us, to all animals and plants, as indicated over
so
here.
the motion is always one-dimensional.
One-dimensional, which is what makes...
What is the dimension is?
That's another fascinating thing about these motors. So you have a cell, right? Well, this room, you can view it as a cell. It's three-dimensional. So where does that one dimension lies? The cells, our cells have a nucleus. Nucleus is inside of the cell. Say, Michael is the nucleus. This scaffold, Dr. Bershine is the nucleus. The scaffold extends away from him so all the structural polarity of minus ends, quote unquote minus ends, starts here. He's the organizing center, and the plus ends extend to the edge of the wall. So the three-dimensionalness of the cell of this room gets to collapse into one-dimensional. So inwards one dimension, outwards another dimension.
Does the scaffold change at all, or is that in this picture it's fixed?
It's static. But it's dynamic. In real life, it's dynamic. It shrinks. There's catastrophes. So these are typically this is blown up, right? But it's walking in the density matched medium.
Is it only the legs of this big blue stuff?
It's
only the legs of this blue stuff.
It's the legs. So.
It's only the legs.
Yeah.
So the brains are there.
The brains are actually here. The quote-unquote brains are over here are the interacting part, the parts that interacts with the scaffold. There's no real brain, in the sense that we don't understand how it walks. But I want to go back to the.
They move to some aim. They want to eat, say, or they want. Every step he takes, you can view it as a conformation change from A to B. Okay? What I'm saying is sort of a very loosey-goosey version. I don't want to be held responsible for it. But you might imagine that in order for me to transform from A to B, I need to consume some energy. And so at each one of the steps is taking a molecule, ATP, triphosphate, breaking apart the bond, and using the energy that's released from that breaking apart the bond, 20 kT, using half of that to make one step, half of that and then break apart another chemical, take another step, break apart another chemical, take apart another step. So if you reduce the concentration of that chemicals, then it waits a while before you can catch a molecule, break it apart, take a step. And waits a while, break it apart, then take a step. And that's how I later on control how fast the motors walk.
You have drawn this as though it's a monotonic thing, but I would think every time you get some more energy available from ATP to ADP, it could go either way. So does it go back and forth?
Not for this motor, but for another one, the notion of universal description is not quite there yet. You can only universally described molecular motors in the sense that it's producing force does work to translocate in mechanical translocation. But in terms of the precise dynamics of each steps, it depends on what you're.
But there must be then some driving force that has directionality.
Oh, so in terms of directionality, one way of describing it is that you can view these scaffolds as one-dimensional lattices, and the lattices has the energy landscape is asymmetric.
Oh, it's asymmetric.
Yeah. So you can pull it one way versus another way, and it takes more force to pull it backwards than to the front or the other direction. I don't remember which one.
And what is the speed?
What's the speed?
At
Saturating at really high chemical concentration, the energy concentration, it goes 1 micron per second, and but you can reduce it down to a lot. But the fundamental question, why the energy landscape is asymmetric, don't you know? Maybe you can tell me. So these are all great questions. These are typically said it's walking on a tightrope in the middle of a hurricane, because it's thermally really at within 1 order magnitude of your KT, which is pretty impressive that it can be deterministic on any level. Although I don't understand this, you probably get the sense that there's not much that I really know, which makes it really interesting. Viruses really quite understand it quite well. Shown here is a section of a neuron where these green dots are herpesviruses. They're exiting the axons by hijacking these motors. Although I don't quite understand this, you can imagine this blue piece as the virus, and then it knows how to control, how to take advantage of these molecular motors. And if you work on viruses, you might think that you might disagree with me, but to me, it's, I'm like, well, I should understand it better than my viruses does. But that's not quite the case. Okay, so now to summarize, these molecular motors are key building blocks of biomachines. They're universal to humans, animals, and plants. They're also they're not universal to bacteria, because bacteria is small, you don't need to translocate for a very long distance. They're also universal specifically to humans, in all cells in humans. And there's one type of cell that I want to focus on in the future is neurons. So this is a cell body, extends in your central nervous system, and then it reaches out to the dismal part of you. So on the person who's really tall, this cell is extremely polarized, and it can be over a meter long. So when you think about a micron per second, these things travel for a micron before it falls off the scaffold. These are very challenging things. Now, because it's universal to all cells in humans, it turns out that just recently people have started to realize that failure in this nano-machine really, inside of the cellular machinery, underlines a number of human diseases. So here are reviews on neuroscience and cancer. And those are two sort of important diseases, umbrella diseases to really fight against, because now we live very, very long ages, and our biomachines start degrading. So how do you prevent it? The biological aspect of my research is if we understand, as viruses do, how to control these motors, perhaps we can reverse some early features, early dysfunctions inside of cells to keep these cells function well and to keep the whole organism function well. And this is not exactly a pipe dream, in that we are understanding more and more in terms of the link between these subcellular nanomachines to the whole organismal failure. When I was in a postdoc, I was really lucky to work with some biochemists and neuroscientists to really identify how defects in that in the other canonical motors can lead to Lou Gehrig's disease. And it was really interesting. In short, you have
each of these, so that canonical motor is made up by 9,000, the basic component, 9,000 lines of code. One line of code in that a mistake of change, quote unquote mutants, leads to late onset neurodegeneration that's similar to that's similar to Lou Gehrig's disease, where these neurons start dying back and cannot make contact with the muscles. Very strange. So anyhow, I don't do that now because I don't have the capacity to work on that. I just want to work on the simple system right now. So these are universal to humans, animals, plants, universal to all cells in humans, and underlies dysfunctions that we call human diseases. Themselves, what we have just covered is really these motors are nano-machines that operate on the energy level of about 10 kT. And my interest lies in not exactly one motor per material to carry, but the question is, when you have more than one cook in the kitchen, what are the collective group function that you can get out of? Is it better or worse, or is it simply linear? We don't know, but we have a lot of evidence that ever since this motor is a canonical motor has been discovered in '85 also, we know that there are more than one that's carrying each what I would call cargo. And there's a whole bunch of evidences on that it's a many-body system. But for a while, people just thought, well, okay, so maybe when you have more than one motor, you just have two motors double the function, three motors triple the function. However, when we look into cells to using microscopies to observe things happening, and this is some of the work that involves myself and colleagues, we see that 1 + 1 does not equal to 2, which is not that surprising for physicists. However, it is interesting. Why shouldn't it be? So here's a physical model that's a predominant physical model that's proposed for the special case that's shown here. That is to say, each of the cargo is carried by more than one motor, but you're carried by identical motors. So you don't have the complexity of one guy going one way and another guy wanting to go the other way, or one guy goes slower than the other, which are also being studied right now. And it's compacted into this equation, which on the left-hand side just says how far this material, this cargo, gets to travel down the road. And I'll return to this. Right now, I just say it was untested for quite a while, until 2012, since it was proposed. And in this talk, I will divide it up into two parts. First is to talk about quantitative test of this theory. Second is to really how to could this theory be these fundamental universal law type of understanding, basic understanding, have any implication for the rescuing or reversing some disease-relevant effects, such as were observed in neurodegeneration.
What is on the right side of this equation?
Huh?
What is on the right side of this equation?
Right-hand side. Oh, so left-hand side is how far the cargo travel. Right-hand side is described as single molecule kinetics. So these, even though in this cartoon,
What does these letters mean
Is
exactly?
that the diffusion coefficient?
So no. So n is how many there are. V is how fast it walks. That you
Velocity.
the velocity, right?
Okay.
Rho is the probability when you have a motor that's in solution of landing on the scaffold, and the epsilon is the probability once it walks. What's the probability of falling off the scaffold? So in this animation, you only see the middle part where it takes deterministic steps. But before that, each of the motor has a probability of interacting, and that is van der Waals kind of interaction, which we can improve the probability of happening using optical traps, and the off rate, the epsilon, is at each step. It has a probability, while it's taking steps, and the step in kinetics, before this one binds, it has a probability of falling off entirely. Of course, falling off in the density matched viscous environment is different from how I make it out to be, but it will just start floating around. It has a probability, again, to rebound. So you can take advantage of these to do simulations.
But can you calculate those probabilities? You don't really know the interactions well.
This one I'll show you so that we can calculate it. It's essentially how far the single one travels, the ratio of velocity to the off rate. That's actually the key of the first part. The probability of interaction is open for the precise number is open for debate, because you can imagine in simulations a higher probability interaction is going to give you entirely different results than the lower probability of interaction. So there is one paper that everyone cites to say this probability is 5 per second, that very few people truly believing, but everyone cites it because it's in the order of magnitude of. So I'm not going to talk about it here, but what we have done in the simulation, which I still have to convince Michael, is to go about a way of getting this row number. And I'll return to this row. It is a free parameter in my system, and I'll return to this row a little later and say that I am just keeping it as a free parameter, and I'll best fit it, and then talk about how we compare with theory.
Excuse me?
Yeah.
I'm confused about the dimension. If n equal 1, then the whole thing is v over epsilon. And what is the dimension of this? V is something
is micrometer per second,
yeah,
and distance over time, epsilon is per second.
Oh.
So v over per second gives you single motor travel distance.
Probability per second.
Yeah.
Okay.
So it's epsilon is the unbinding time per unit time, and this is d. The little d will be spatial association time. So I'm a physicist. I don't know if you, by training, I don't know what I am now, but I don't ever want to go into a cell and then purify these things. Purify meaning isolating these things, because it actually is a quite untrivial art. So I rely on my awesome biochemist friends. They give me these test tubes. So when I say goodbye, I will be very sad. These test tubes are purified, isolated, just this motor amount, or just these cytoskeleton scaffolds. This is a very non-trivial, but I will gloss it over. And the method is you have seen, I'm training in the same lab as Michael Verschin, and we use optical trap. And optical trap is really started the moment that laser was first invented by Arthur Ashkin. And first thing was levitating using optical pressure. And this is a seminal paper in optical lasers. I think it's cited 4,000 times, something ridiculous number, but it's continually growing. So physicists have really, so this paper has Stephen Chu on it. Physicists have really ignited and shaped the single molecule field. Optical trap is very hard to work out the actual physics of it. It's because in our particle size regime, which is a meter regime, so you cannot do any sort of approximation. However, it's very easy. And at the end of it, I asked myself, what sort of intuition did I gain? The answer is not very much. So I like to use ray tracing. Basically, at the focal point of the light, you have lights that are going in each and every direction. When you incident on a dielectric sphere that has higher index reflection than the surrounding medium, light will bend. And depends on, so if the index refraction is higher than the surrounding medium, it bends in one way and then otherwise bends another way. And you can, I can convince myself that the momentum that's imparted by this refracted light is going to give me a net force that is pointing toward the most intense part of the focal point, which is a Gaussian distribution. So I'm happy staying there. And I'll return to so optical trap has been really used to study one particle. So this one dielectric sphere is being used to both exert force on the motor that is associated with, and also for visualization. And it's been used mostly until recently strictly in the single molecule case, that is to say one motor, one cargo, one-to-one interaction. And there's still a lot of great science that's coming out of it. Recently is by the Merced lab. And not to leave myself out, I will say, oh, well, I also did some single molecule stuff. It's interesting because this paper is on, both of these papers are on this one canonical motor called kinesin, for those in the know. And this paper is on how to turn off this kinesin. And this paper is on how to turn on this kinesin. So if you imagine your espresso machine, there's an on button and off button, right? And we're not very accustomed to think of protein machines as having on and off button, but there should be. Otherwise, why should it be different from this macroscopic analog? But when you have more than one motors carrying the same cargo, the physics of optical trap remains the same, the imaging remains the same, then you can start thinking hard about how to back out the dynamics between different identical motors for this special case. One of the challenges is using optical trap here is actually for multiple motor study is to how to control this number of motors. Because each of these V, rho, and epsilon that I will return to, there are single molecule descriptions. To some extent, that can be confined, the range of value can be confined, although that's still a challenge. So how do you control this? Number of motors. Turns out that everything that should be done has already been done. In early 1990s by Arthur Ashkin, we already know that motor force production is quantized. That is to say, if you have one motor on the cargo in optical trap, you'll see this type of force profile. I call it RC shape, so you have a fast rising and a plateau. And if you have two motors, you have two level plateaus. So by simply counting the force, you can count the motors. So you can, this is non-trivial to do, but it's doable. And so the first project is really to quantitatively test this theory, again using very simplified components. The cytoskeletal tracks, we fix them, and then the optical trap to bring a bead, dielectric bead with motors, more than one motors coated on it, and then bring it close, bring these two things close to each other so that the distance for interaction is not so great. You can greatly increase your data statistics. And the idea is to test this. The first thing that we did was to make it as simple as possible. That is to say, let's set n equal to 2. 2 is the simplest case for more than one motors. And that is pretty easy because I just replace n with 2 and then rewrite it in the simplified version. Here's the more spelled out version of the rho and epsilon and the velocity. It has a probability when it's in solution far away from the cytoskeletal track. It has a probability per unit time to interact with this. It's like my scaffold is over here, and this guy is all over the place, and it binds per unit time. Stochastically happens. Once he binds, he has a probability of rate of taking the steps, which we can control using the chemical concentration. And at each step, he has a that is to say, at each unit time, he has a probability of dissociating and separating this interaction again. So this is all good and well, except that as some of you may recognize that these are not, although it's clean looking, it's not necessarily true that any of these parameters should be independent. And they're not. The velocity, how fast it walks, and its per unit time of dissociation rate becomes, is intertwined. That is to say, this motor, every step it takes, it experiences a probability of falling off. When it doesn't take steps, it remains anchored down. So that's a problem, but it's not that big of a problem because we know how it relates to each other. So micrometer per second, this is how far a single motor will travel. And I can measure this. Yeah?
Why should the probability of falling off be any different at one site than at another site? I thought you said that probability of falling off could increase, and yet I would think the probability per second or whatever falling off would be the same, because one-dimensional symmetry here on this dipole.
I mean, we'll go in one direction versus. So this is a nice motor in the sense that I don't have to worry about it stepping back. It just steps forward.
Right.
And every time it steps, it has a probability of falling up.
Right.
So,
So the only reason that might change is because the energy is going down, right, or not? Is there?
that is a far more complicated question than I can possibly even try to start answering. So here I'm taking advantage of a phenomenological. So this equation, even though it's back of the envelope, is from, I didn't cite it, but over a decade of study to reduce down to this one simple equation. But it's very phenomenological. Exactly why and the hows in each step, I cannot. I do not actually know. Is that so? So your question is not answered.
It's okay.
Okay.
You did a good job.
So at some point, I have to make peace with being phenomenological, but perhaps sometime down the road, these are, because there are tons of people who are answering these questions. So because it's not a problem because we know the relationship. Once we know the relationship, we can plug it in and recast this theory into something that's have independent parameters. So the velocity and the probability of interacting is completely independent, and the velocity and how far it travels is completely independent. And I'll show that control. I will demonstrate that using data in a little bit. Okay, so once we have this, we say, okay, this looks pretty bizarre because he says that when you have two motors, two identical people carrying the same couch, walking on a tightrope, and depends on how fast and how slow they walk. You might imagine that they walk better or worse with a different speed, but certainly not going to be inverse of this velocity.
Traffic is the name of the journal?
Huh?
Traffic is the name
Traffic.
of the journal?
Oh, excellent question. So when I talk to physicists, they always ask me traffic. So it's a biophysical journal. This paper, since it was published in late 2012, was actually cited for like 12 or 11 times, depends on how you count it. So, but it is unknown to physicists, and it started in 2008 or 7 by. Huh?
It's very known in Sabah.
I get, yeah. So traffic. I have a list of journals that I would like to publish in that have funny names, but okay. So the prediction is that the two-motor travel is going to be inversely related to velocity, and you can imagine that if I reduce, this says that it's really a large tunability. Right? If I reduce the velocity by a factor of 10, the travel distance should go up by a factor of 10. That's a little bizarre. I can imagine variations in a factor of 2 also. So let's measure it. But first is sanity check. First is to say, all right, do I measure, using my setup, do I measure what is known on the single molecule case, which is at saturating, giving a lot of chemical energy? Do I see approximately the right velocity? The answer is yes. And do I see approximately the right distance, single molecule travel distance, the spatial association rate, time? And when I have two motors, what do I see? Roughly the same velocity as it should be. And so these are histograms, counts per distance, and you can fit them to a single exponential for the distance and the Gaussian for velocity. And you get two motor travel under regular conditions is about 1.77 times the single motor travel. 1 + 1 is less than 2. Not surprising at all because in entirely different setup where Michael Dale's group at Rice in 2009, they already came on and said, hey, when I physically link two motors together with a DNA double strand, the persistence length is 50 nanometers. So it's really far spacing. It's a different geometry. They get 1.73. So pretty reasonable agreement, and we'll move forward. What they didn't do is just say, well, how does the two groups travel differ at different velocity, which is what we're trying to measure. So what you're going to see here is fast velocity, slower velocity as seen by the central peak, and even slower velocity.
on a single molecule level, this little d remains constant. The histograms remains roughly the same with the same decay constant. On the two motors, the travel distance at first is moderately increased, and then this large bar over here is my field of view. All the cargos, the dielectric beads, just walk out of, like my head, walk out of the field of view already. So I just count them. You can see that there's greater and greater fraction of the two motor travel out of my field of view.
Excuse me, please.
Yeah.
If there are two motors, they probably should know about each other, otherwise they risk to lose their cargo.
So I biochemically link them together. Now, whether or not they know so in this setup they shouldn't know each other. But I'm not going to talk about this in the modeling that we're doing. They do interact. The behavior of one depends on its partner, which is I think I actually have some physicists as my reviewers, but which is surprising because they are independent of each other. They shouldn't know each other if they do. You can argue that perhaps modifies the energy landscape of the microtubules to change the binding unbinding, but I don't know.
Are they competing for fuel?
No, because at this level, there's enough fuel, so you can still you can reduce the fuel down to an order of magnitude smaller than this, and at that point you start becoming a starving issue. But then you can put in recycling systems to put molecules back together.
But you don't study them when they're starving.
No. But this, my prediction is that if you go to really, really low level, you will never see any dissociation of the cargo from the scaffold. Of course, it's not quite moving, but so with this, here comes the comparison to theory part, and returns to your question of what the rows and so on values are. I don't know what my on-rate probability of interaction is, but I can fit it and find the best fit value, and then I make prediction using this constraint theory. I can make some predictions at different velocities and compare the constraint predictions with the measurements as we deviate from the point where we fix our theory. So this is one technique that can be more successful in biophysics type of comparison because you just don't know everything. So you constrain your theory and see how well it performs as you deviate away from that constraint. And these are two deviation points, although it's just 2 points, but it's two deviation points. And the prediction, which is light gray, and the experiment agree to a fair amount of, agreement. And really the first quantitative verification of this prediction, the takeaway really goes to this spatial unbinding rate and temporal unbinding rate. This is probability per unit time that each motor, in the absence of any interaction, each motor will fall off the microtubule or the scaffold. And because the single motor travel distance, this little d remains constant. This is constant. As the velocity reduces, the unbinding rate per unit time will also reduce. So per unit time, if you each motor that's bound to the scaffold will have a greater probability of acting as just an anchor that links the cargo with it and buys more time for the next motor that's unattached to become attached. Then if it's become attached, then the cargo cannot dissociate away from the microtubule, the scaffold. Then you increase the association time, and then for a certain velocity, the travel distance increases.
And the cargo has some special device that keeps it on track.
The cargo is on the track, remains on track so long as these legs remain associated on the track.
Well, look, when a man is moving on two legs,
Yes.
it's pretty difficult, I would say. It's pretty difficult work of our brain to keep us
Yes.
not falling.
Yes. But I am also not eating bread every single step. These are chemically stimulated conformation changes. Every step it takes is driven chemically, energetically driven.
So how do you supply ATP?
Oh, I just put it in the concentration, in the solution. There's no magic to it, it's just sugar in water. So a good analogy is that I'm not drinking one sugar molecule per step.
In living organism, then you have ATP produced by creatine kinase or glycogen solution, right? But here, if you put sugar in there, the sugar doesn't produce ATP by itself.
Oh, no, I put ATP in the solution. Yeah, I'm making a careless analogy.
So, okay, so you put enough.
So the fast ones over here are at 1 millimolar ATP. Here is 20 micromolar ATP. Here is 10 micromolar ATP. Yeah.
So with these velocities, I mean, it looks like when you reduce the velocity, you can walk a lot further. So how do these velocities compare to what is observed in Vivo?
Yes.
Which one do you see?
Super good question. Although I'm actually not able to answer that quantitatively, other than saying we were very excited to finally say that velocity has some is of interest, because before it was just okay, that's the rate we can control. But if you look into cells, there is a wide range of motion. So if you look into a neuron, there is slow transport, slow axonal transport, fast axonal transport, and there's to a large extent has been just phenomenologically classified as that. And this brings up a possibility that at the cost of going slower, you can reach further distance. Another thing is that, say you're under ATP-deprived conditions, say ATP is produced by mitochondria from my popular science understanding. In a stroke, say ischemic stroke, when you are oxygen deprived and all sorts of terrible things happen to mitochondria, you have ATP-starved conditions for these neurons. So under those conditions, of course, I'm just extrapolating wildly, that this may be this velocity, slower velocity, because you have ATP-starved condition, slower velocity may be one mechanism, that building mechanism for the cell to continually having some transport under the stress environment. Now, exactly what the ATP concentration is in neurons, I think the lower end is 100 micromolar, not as low as what I have here. But I wanted to make a point here versus I want to see a large distance. So at some point I will measure what 100 micromolar, what the tunability is. It's going to be within a factor of 10. Yeah. So, and this is this whole notion of buying more time. It's just like you're walking along a monkey bar, right? You better have at least one motor bound to the monkey bar before the other one catches up. And it's generalizable to more than one or more than two motors. Okay, so I will have one more minute. I will just quickly breeze through the next thing, which is what happens when we block the scaffold. It's a beautiful, nice, fresh highway, but let's put lots of potholes on them. These potholes are important because you've heard of this word tau. Well, you've heard of the word tau, but in a biological context, has to do with some sort of potholes that you find in Alzheimer's disease. And we study using the same assay, same experiments, except that we just coat the scaffold with these tau, again supplied by our biochemistry friends. And so here's a macro scale analogy of what I see motors. This is a person, but because it's not density matched, he has a probability of falling off. So he has some balancing thing going on. But it's taking steps. Every step, he has a probability of falling off, right? We're not demonstrating the on rate. So what this tau does is really you have another guy that's lying here, because there's another association dynamics that's being studied along here. And I'm not showing the video because this guy successfully steps over and remains on track, but he also is a world record holder. So this tau, what it does is in terms of single molecule kinetics, is reduce this interaction rate. It reduces the probability of interaction, increase the probability of dissociation. And I'll point out that the first one who really definitively showed this was Michael Verschunen. And people have suspected this because when you have this picture, you say, okay, yeah, of course. But people have suspected this, and people have tested it, big groups, and they did not see an effect because there are a number of things that you have to look out for these experiments. But since he identified this, there's a whole bunch of studies that came out supporting it. Now, then this is straight out of Michael's paper, where again, histogram of travel distance. You have when there's no potholes, clean, beautiful highways, everything walks out of your field of view, as indicated by this bar. When you have potholes, your histograms of distribution gets collapsed back to some finite distance that you can quantify within your 8-micron window. So the prediction is that if I slow down these motors, I'm going to recover the travel distance. And feel free to walk away. So here is some video recordings. It's very close and dear to my heart. Now, what you're seeing are these tiny little beads, dielectric beads that I can visualize based on its index refraction. They're carried by more than one motors, and they're walking insanely long distance along these faint microtubules. Insanely long, I mean 20 microns. And I don't even need to use optical trap to bring the thing close to the scaffold. The interaction probability is high because there's a lot of motors on it. What happens when I coat the microtubule, the scaffold with potholes, tau? I now have to trap a bead in my center of field of view. I don't know the polarity, structural polarity of the scaffold, so I have to put it in the center and just let it go one way or another. And you see, it's with the remaining in the center mass, the field of view, and then it disappeared. The reason it disappeared is because it dissociated from the scaffold and it floated away. When it floats away, this is the interferometer, you no longer see the specific type of pattern. What happens when I slow it down? Play all three of them so you can see that the velocity is low. But you can see that I'm no longer needing to trap it. There is self-assembly over here. This guy already fell off. It's going on, and there is self-assembly over here, and it just keeps on going, keeps on going, keeps on going. And in the interest of time, I'll just leave this. And you can quantify the respective travel distance. This is control sanity check to say, okay, yeah, my pothole is really doing what it's doing, reducing the travel distance, multiple motor travel distance. And by slowing it down, I'm actually recovering. Not as extreme as I predicted, but that's okay. Let's do it a few times at different motor per beat concentration, just as indicated by the overall natural travel distance along the beautiful highway. And you see this the same phenomenon over and over again. This part, the top two panel, is sanity check to say, okay, my pothole really is doing what it's doing. It's reducing the travel distance. The bottom two is new data, which is in biophysics later. I went away from traffic because I wanted my students to know that I'm actually doing biophysics. Yeah, but anyhow, so you can see that there is an increase in travel distance in the average of this distribution, right? And that is the new finding, that despite the presence of a pothole, you can recover the travel distance. You might imagine that in a polarized long cell, such as a neuron, the ability to maintain travel distance that you cover might be an important thing. And the take home will be the velocity. Velocity can be a promising route to reverse the dysfunction in diseases, such as in Alzheimer's disease. So I'll just conclude with my awesome undergrads. He's going to leave me soon. And a few undergrads and a rotation student. And I think Yosemite is around here. I'm collaborating with a random walk theorist here. This is my wonderful, wonderful biochemist collaborator. This is the person who trained me to work with molecular motors. If you're looking for a job, here's my number. So thank you for your attention.
So, very nice talk. The one thing is how important it is for it not to fall off. I mean, if it's traveling on the axon, and imagine that it falls off 30 centimeters into an axon, in that small axon, it's going to have a really high probability of rebinding, you know, and keep going. So, I mean, how does it, you know?
I don't know. So in the Lou Gehrig's disease, that study, when we look at the mutation to the dynein motors, on the single molecule level, you only reduce the travel distance by 20%. And on the multiple motor level, inside of a cultural neuron, that 20% amplifies to 50%. And that 50% in the mice animal, the cultural neuron is from mice, not by me. You can see a phenotypical effect. That is to say, the muscle control is not there.
Yeah, but that's a mutant, no?
Yes.
So if you talk about the mechanism, I mean, if the mechanism is supposed to be try to get from A to B in the fastest possible way,
Maybe
and the
fastest.
and the most reliable way, you can have two ways. You can go very slowly and try not to fall off, or you can go very fast and fall off and fall in a rebind and go very fast again.
So,
which one would win over the long distance? Have you done that analysis?
so when you rebind, okay, so that's why. So in this, I will return to the mice model of the Lou Gehrig's disease. At first, in 2004, one paper came out and saying that the vesicle transport, the velocity is what's important. Just by slowing it down, makes a difference to the whole animal activity, what have you. But what we found is that it's not the velocity. Velocity remains the same. It's a premature dissociation. Once it's dissociated inside of this tunnel, this cargo is confined. It doesn't quite have places to diffuse around, but diffusion doesn't work anyways for these large particles, and it's confined, and so its probability of pausing becomes significantly higher. So it's going at the same velocity, but it pauses. So when you're imaging at a coarse frames per second, you think it's slowing down. How important is it? I don't know, but you might be able to. Let me speculate. If there is only one motor, perhaps one type of motor, perhaps not that important. However, if you have different motors, just two different canonical motors, one going one way and another going another way. When you reassociate, if there is a difference in reassociation time, you might go back. Who knows? I don't know. So that's one argument that I will make. I don't honestly know how important travel distance is. It's just it is one well-defined measurables for this black box.
It's definitely very interesting that when you slow it down, it was very
bizarre, right?
interesting
Yeah,
observation.
and it's totally predicted in theory, and
But theories are always wrong.
well, as you
Oh, no, I'm sorry. Oh,
hey, I'm an experimentalist, I totally agree.
The tight part goes fast and falls off.
But Steve and I spent at least a good week yelling at each other on whether or not that prediction is right, and then in the end, I just said, let's do it. Because
Now, in plants, there's this phenomenon of protoplasmic streaming, which I'm sure you're familiar with, which kind of looks like this as a transfer of food or whatever from one part of the plant to another. This is not at the molecular level, it's at a much larger level. I was wondering how much of your thinking would carry over to problems like protoplasmic streaming.
I don't have a clue. I'm not even outside of an animal cell. I may be confined to 500 nanometers around this bundle of microtubules. But I will certainly go home and look up those plasmic streaming and try to figure out something. Yeah.
So in this terms of being less likely to dissociate when the speed is lower, is that, like, can we think of that as an energy effect in terms of the faster step being a more energetic step and something that's more likely to kick you out of a given semi-well-defined potential, or?
I think so. When you can think of the kicking out of the association state as happening when you transition from state A and state B, A and B. So in terms of that, but the energy conservation part perhaps is, I don't know if I really can make any statement out of it. In that so
Is there a kinetic energy to those steps that's important to whether or not they succeed or not, or is that?
I think the answer is yes, and I don't know how the kinetic energy. I mean, again, I don't know how the details of stepping really, really works. I believe you have something new coming out of the Verschunnen's lab that can give you some. So they look at variable temperature measurements per steps. That might give me some indication of, because a number of things happening in the steps. You have to grab onto a molecule, break it apart, and that happens under the hood of this foot that's associated of the molecule, under the hood of the car, and break it apart and spit out something and holding onto the diphosphate molecule. That's one conformation. And then you have to somehow evacuate that, and then during that process, there is a change in ATP constant. So exactly what is the rate limiting process, I don't, in that whole sequence of events, I don't know. I simply group them into one step without diving into the details. But maybe he can tell you more with his stuff.